The human sense of smell is one of the most evolutionarily ancient and behaviorally influential systems in the brain. What sets olfaction apart from other senses is the directness of its neural pathway. When we encounter a scent, say freshly cut grass, perfume, or the odor of smoke, those volatile chemical molecules enter the nasal cavity and bind to specialized receptors in the olfactory epithelium. Each receptor is attuned to specific molecular features, and this binding triggers a cascade of electrical signals that travel directly to the brain. Here's where things become fascinating from a neuroscience standpoint. Unlike other sensory systems, olfactory information bypasses the thalamus, which is typically the brain's relay center for processing sensory input. Instead, these signals travel directly to the amygdala and hippocampus regions deeply involved in emotion, motivation, and memory. This unique architecture explains why scent is so profoundly tied to emotional memory. You can likely recall moments where a particular smell may be your grandmother's kitchen, or the cologne of someone you once loved, instantly transported you back in time, triggering emotions and sensations more vividly than a photograph or song ever could. From a neuroscientific perspective, this is not merely poetic, it's hardwired. The olfactory bulb, the brain structure that processes smell, is part of the limbic system. It has direct synaptic connections with areas that govern primal behavioral responses. This is not just true in humans. Across mammals, smell is deeply linked to survival behaviors, finding food, detecting danger, identifying kin, and selecting mates. To make this more concrete, consider how olfactory cues regulate behaviors in animals. In rodents, for instance, a single molecule emitted in the urine of a dominant male can alter the reproductive behavior of a female or suppress the fertility of subordinate males. These changes are not the result of conscious awareness but automatic, hormone-driven responses. The same principles apply to humans, although we often underestimate the role of scent in shaping our behavior, because these effects tend to operate below the level of conscious perception. Studies show that exposure to certain human body odors can alter cortisol levels, indicating changes in stress, and even shift perceptions of attractiveness, trustworthiness, or dominance. This gives rise to an important concept in behavioral neuroscience: unconscious sensory priming. The air around us is filled with molecules we cannot see, often cannot name, and sometimes cannot consciously detect, but which are nonetheless impacting our physiological state and behavioral responses. Consider the influence of ambient smells in spaces like hospitals, schools, or homes. The smell of disinfectants, food, or body odors individually or in combination can shape our sense of safety, comfort, alertness, or even our ability to concentrate. One fascinating area of current research focuses on how specific scents modulate brain states. For example, certain essential oils like lavender have been shown in functional MRI studies to reduce activity in the amygdala and hypothalamus areas involved in stress response, leading to measurable reductions in anxiety and improved sleep. On the other hand, peppermint oil or citrus-based scents can enhance alertness by increasing activity in regions involved in attentional control. This is because olfactory inputs are not just being processed passively. They're actually interacting with neuromodulatory systems in the brain, such as the cholinergic and noradrenergic systems, that regulate arousal, mood, and executive function. Let's now link this to neuroplasticity, the brain's ability to change in response to experience. Because olfactory inputs can influence emotional salience, they can also tag memories with increased importance. In other words, if a particular event is paired with a strong smell, especially one that has positive or negative valence, it becomes more likely to be encoded deeply and retrieved vividly. This has implications for learning, therapy, and behavior modification. For example, incorporating scent into therapeutic environments or even educational settings could enhance retention and emotional regulation. This is not speculative. In animal models, odor cues have been used to reactivate memories during sleep, increasing the consolidation of those memories in the hippocampus. What's even more compelling is how smell modulates social behavior. Humans are constantly emitting chemosignals through sweat, breath, and skin. These signals are often laden with information about emotional states. A classic study demonstrated that sweat collected from individuals in fearful states, such as during skydiving, can increase amygdala activation in people who smell it later, even if they are unaware of the source. This kind of unconscious emotional contagion is a key feature of how we interact socially. You might walk into a room and sense tension, not just by observing body language, but because your brain is literally picking up on the chemosensory profile of stress hormones being excreted by others. Another domain where olfaction plays a pivotal role is in appetite and feeding behavior. The hypothalamus, our brain's command center for hunger and satiety, is tightly coupled with olfactory processing centers. In fact, the smell of food can trigger anticipatory insulin release, even before you take a bite. This anticipatory response, sometimes called the cephalic phase, is crucial for digestion and energy balance. It's also the reason why exposure to certain food odors can make us feel hungry even when our stomach is full. In people struggling with disordered eating, such as binge eating or anorexia, olfactory sensitivity is often altered, suggesting that recalibrating sensory cues may have therapeutic benefits. We also need to consider developmental aspects. Infants are born with a highly sensitive sense of smell. Within hours of birth, they can recognize their mother's scent, which guides bonding and breastfeeding behavior. This olfactory imprinting is foundational to human attachment, and disturbances in this early sensory bonding, such as in preterm infants separated from caregivers, can have long-lasting effects on emotional development. Similarly, individuals with impaired olfaction, due to trauma, age, or infection, often report decreased enjoyment of life, increased depression, and impaired social connection. This highlights how essential the olfactory system is, not just for survival, but for psychological well-being. Let's bring in one more key dimension: plasticity and recovery. The olfactory system, unlike most other sensory systems, has a remarkable capacity for regeneration. Olfactory receptor neurons can regenerate throughout life. This makes it a potential model for studying neural repair and recovery. For people who have lost their sense of smell due to viral infections or head injuries, olfactory training, repeated exposure to specific scents, can help retrain the system, rewiring the brain's processing networks. This training doesn't just restore smell. It often improves mood, memory, and cognitive clarity. In conclusion, the science of smell is not just about odors or preferences. It is a direct portal into the deepest parts of our brain. Smell shapes emotion, memory, social interaction, motivation, and even cognition in ways that are often outside of conscious awareness. By understanding how the olfactory system works and how it connects to neural circuits responsible for behavior, we gain powerful tools for improving focus, regulating emotion, enhancing relationships, and even accelerating recovery from trauma or illness. Whether through therapeutic use of scent, design of environments, or personalized behavior change protocols, harnessing the influence of smell on the brain is a frontier in both neuroscience and human performance. Taste is far more than a simple matter of preference or flavor. It is one of the most behaviorally instructive sensory systems we possess. The gustatory system, which governs our perception of taste, is intimately wired into the neurobiology of survival, decision making, reward, and emotional learning. The basic categories of taste, sweet, salty, sour, bitter, and umami, each correspond to specific survival needs or potential dangers, and each one activates different circuits in the brain with immediate consequences for behavior and physiology. When we taste something sweet, what we're detecting is typically a signal of energy, rich glucose or fructose, which our bodies need for fuel. This detection begins in the tongue, where taste receptor cells housed in taste buds bind to sugar molecules. These receptors, known as G-protein, coupled receptors, then send signals through the gustatory nerve to the brainstem, then to the thalamus, and ultimately to the primary gustatory cortex. From there, signals are relayed to deeper brain structures, particularly the insula, orbitofrontal cortex, and limbic areas like the nucleus accumbens and the hypothalamus. Here's what's key. The experience of sweetness activates dopamine release in the mesolimbic reward pathway. This circuit, which includes the ventral tegmental area and the nucleus accumbens, is the same circuit that's activated by other forms of reward, including social approval, monetary gain, and drugs of abuse. From an evolutionary standpoint, the pleasure associated with sweet taste reinforced behaviors that would lead to the acquisition of calorie-dense foods. And the reason this system persists so strongly in modern environments is because the brain does not distinguish between evolutionarily rare sources of sugar, like fruit, and modern concentrated ones like soda. Both trigger powerful reinforcement and drive continued seeking behavior. Salty taste, on the other hand, serves a fundamentally different but equally important purpose. Salt, or sodium chloride, is essential for neural conduction, muscle contraction, and fluid balance. The craving for salt is deeply embedded in the hypothalamus, which regulates homeostatic drives like hunger, thirst, and thermoregulation. When sodium levels in the body drop below optimal, specific neurons in the hypothalamus detect this and increase our drive to seek out and consume salty foods. Again, this drive is not purely conscious. It is a fundamental motivational state driven by internal chemistry that guides external behavior. Now consider sour and bitter tastes. Both of these are often signals of danger in nature. Spoiled foods, unripe fruits, or toxic compounds tend to be sour or bitter. The gustatory system is wired to respond to these stimuli with aversion. In fact, bitter taste receptors are the most genetically diverse of the taste receptor families, likely because detecting a wide range of bitter compounds conferred an evolutionary advantage by helping our ancestors avoid poisons. Importantly, the amygdala, a brain region involved in threat detection and aversive learning, becomes highly active during exposure to bitter or overly sour tastes. This creates a negative reinforcement loop. We learn to avoid these tastes in the future because of the discomfort or nausea they may produce. Umami, the fifth basic taste, detects glutamate, an amino acid found in protein-rich foods like meat and cheese. The detection of umami is associated with protein valuation and muscle repair, particularly important for growth and reproduction. Just like with sweetness and saltiness, umami activates reward centers in the brain, reinforcing the consumption of protein-dense foods when the body needs them most. Interestingly, the preference for umami increases under conditions of physical stress or growth, indicating that the brain can recalibrate taste preference in accordance with physiological demands. Now, let's shift from basic taste perception to how these taste signals guide complex behavior and decision making. The experience of taste is not merely chemical or sensory, it is tightly integrated with learning and memory. When we consume a food and have a positive or negative outcome, say increased energy or food poisoning, that association is encoded in the brain and influences future behavior. This process is governed by the insular cortex, which integrates taste with interoception, our sense of internal bodily states, and by the hippocampus, which encodes contextual memory. The result is that taste becomes a teacher. It helps us learn what to seek, what to avoid, and under what circumstances. Let's take a moment to consider how powerful this mechanism really is. Imagine you eat something unfamiliar and later feel nauseous. Even if it wasn't the food that made you sick, your brain will often associate that food with the discomfort and create a strong aversion. This is called conditioned taste aversion, and it can occur even with a single exposure. It's one of the most robust and rapidly formed associative memories the brain is capable of creating. This has been demonstrated in lab animals and humans alike. The reason for this heightened sensitivity is evolutionary. Avoiding a potentially toxic food can be a matter of life and death. Now we turn to how taste interacts with the brain's reward system and how that shapes our choices over time. When a food is palatable, that is, when its taste is rewarding, it activates the orbitofrontal cortex, a region involved in assigning value to stimuli. This value is not fixed. It depends on a range of factors: nutritional status, emotional state, prior experience, and social context. For example, the same food may taste better when you're hungry than when you're full, or more enjoyable when eaten in company than when eaten alone. The orbitofrontal cortex integrates all of these variables to determine the motivational salience of the food, that is, how much you want it right now. Here's where things get really interesting from a neurobehavioral perspective. Modern food environments often hijack these reward circuits. Highly processed foods combine sugar, salt, fat, and flavor enhancers in ways that create what's known as supernormal stimuli, exaggerated signals that elicit stronger than natural dopamine responses. These responses can drive compulsive eating, independent of hunger or nutritional need. In animal studies, rodents will work harder to obtain processed, sweet, fat food than even addictive drugs like cocaine. In humans, this manifests as craving, loss of control, and dysregulated behavior around food. There is also an important cognitive dimension to taste. The brain's prefrontal cortex, responsible for planning, impulse control, and future-oriented decision making, is engaged during food choice, especially when we attempt to override taste-driven impulses. For example, when you choose a healthy but less tasty meal over a more indulgent one, your prefrontal cortex is working to suppress activity in the more primitive reward centers. However, when you are sleep deprived, stressed, or emotionally dysregulated, the prefrontal cortex is less active and the reward circuits dominate, leading to poorer food choices. This explains why emotional states and stress so strongly impact eating behavior. The neurobiology of taste is embedded in larger networks of emotional regulation and self-control. It's also worth mentioning the role of expectation in shaping taste perception. Placebo effects are well documented in the realm of medicine, but they also occur with food. When people are told that a wine is expensive or that a food is healthy, their brain's reward circuits light up more strongly, and they report higher satisfaction, even if the actual food is the same. The brain doesn't passively receive taste information. It actively constructs it, influenced by beliefs, labels, and prior experiences. This gives rise to powerful strategies for behavioral change. By altering how foods are presented, framed, and contextualized, we can reshape how they are experienced and ultimately how they are chosen. Taste is also deeply embedded in social behavior. Eating is a profoundly social act, and the gustatory system is influenced by group dynamics, cultural norms, and shared experiences. Mirror neuron systems in the brain may play a role here. Observing others enjoying food can increase our own desire for that food, a phenomenon known as social facilitation. Conversely, witnessing disgust or aversion in others can decrease our appetite, even if the food is otherwise palatable. These responses are thought to be mediated by circuits in the anterior insula and anterior cingulate cortex, regions involved in empathy, pain perception, and shared emotional experiences. Finally, taste is modifiable. Like smell, taste preference is not fixed. Through repeated exposure, changes in internal states, or conscious reappraisal, the brain can learn to enjoy foods that were once disliked, or to lose desire for foods that were once craved. This plasticity is a powerful tool for behavioral intervention. For example, gradual reduction in sugar content of foods over time can lead to a recalibration of sweet preference, reducing overall sugar consumption without creating a sense of deprivation. Similarly, mindful eating practices, which involve paying close attention to the sensory experience of eating, can change how the brain encodes taste and reduce impulsive eating. In sum, taste is far more than a sensory experience. It is a powerful driver of behavior, emotion, memory, and decision making. The gustatory system is deeply embedded in the brain's motivational and regulatory architecture, guiding choices that range from the mundane to the life-altering. Understanding the neuroscience of taste allows us to leverage it for better health, clearer cognition, emotional balance, and behavioral resilience. Whether through deliberate exposure, environmental design, or mindful engagement, we can use our understanding of taste not only to eat better, but to live better. The concept of pheromones, though often misunderstood or sensationalized, represents one of the most powerful and underappreciated aspects of human and animal behavior. Pheromones are chemical signals that are emitted by one individual and detected by another, producing a specific and often unconscious change in behavior or physiology. These are not just scents, and they are not experienced like the smell of coffee or perfume. They are molecular messages encoded in chemicals that bypass cognitive processing and influence neural circuits in ways that are deeply rooted in evolution. In animals, the role of pheromones is well established. They regulate social hierarchy, reproductive behavior, territory marking, maternal bonding, aggression, and even synchronization of group behaviors. A single molecule emitted in the sweat or urine of an animal can suppress the fertility of others in the group, alter stress levels, or even trigger mating behavior. These effects occur without the need for conscious thought or decision making. The detection of pheromones occurs through specialized chemosensory structures, including the vomeronasal organ VNO, and their signals are routed directly to brain regions responsible for hormone regulation, instinctual responses, and emotional behavior. In humans, the presence and influence of pheromones are more subtle, but emerging research shows that they are no less significant. While the functionality of the vomeronasal organ in adult humans remains debated, the human olfactory system, particularly through the main olfactory epithelium, is capable of detecting and processing a wide range of social chemosignals. These chemical cues, emitted through breath, skin, and bodily secretions, convey information about an individual's emotional state, immune profile, genetic compatibility, reproductive status, and even social intentions. One of the most studied examples in humans involves the synchronization of menstrual cycles among women living in close quarters. Though controversial, some studies suggest that exposure to axillary secretions, sweat from the underarm area, from one woman can influence the timing of ovulation in another. While the exact mechanisms remain under investigation, the implication is profound that human reproductive physiology can be modulated through airborne chemical communication, operating below the threshold of awareness, but the influence of pheromones extends far beyond reproduction. For instance, experiments have shown that sweat collected from individuals in specific emotional states, such as fear or happiness, can alter the behavior and brain activity of others exposed to. In one study, participants who smelled fear, induced sweat displayed heightened vigilance and activation in their amygdala, the brain's threat detection center, even though they were unaware of source or nature of the scent. This suggests that humans are constantly and unconsciously broadcasting emotional signals through their skin, and others are picking up on these cues and adjusting their emotional states accordingly. From a neuroscience perspective, this represents a remarkable form of nonverbal, non-visual communication. The olfactory system interacts directly with the limbic brain, the structures responsible for emotion, memory, and autonomic regulation. That means chemosignals can change your heart rate, shift your cortisol levels, or alter your attention patterns without any conscious recognition of a change in your environment. You might feel uneasy around someone or inexplicably comfortable in another's presence, and while you may attribute it to body language or tone of voice, it could well be the influence of undetected pheromones. The implications for social behavior are vast. In humans, pheromonal cues likely play a role in mate selection. Research shows that women tend to prefer the scent of men whose immune system genes, specifically, those encoded by the major histocompatibility complex MHC, are different from their own. This preference promotes genetic diversity and potentially healthier offspring. Interestingly, the use of hormonal contraception can reverse this preference, which might explain changes in attraction or relationship satisfaction that some women report after starting or stopping birth control. There is also evidence that pheromones influence perceptions of dominance and submission. In studies where participants are exposed to the body odors of men with high testosterone levels, they tend to rate those men as more dominant, even without seeing them. Similarly, exposure to chemosignals associated with stress can lead to elevated stress levels in observers, mediated by increased activity in the hypothalamic, pituitary, adrenal HPA axis. These effects are not just physiological curiosities. They translate into real behavioral outcomes, from increased alertness in potentially threatening situations to subconscious biases in social judgment. Another area where human pheromonal communication has a major impact is in parent-infant bonding. Newborns are extremely sensitive to maternal odor and can distinguish their mother's scent within days of birth. This scent recognition facilitates nursing, soothes distress, and helps build early attachment. Mothers, too, respond differently to the scent of their own infant compared to others, with changes in brain activity observed in the orbitofrontal cortex and limbic system. These responses appear to be part of an evolutionarily conserved mechanism for enhancing care and protection of offspring. What's important to note here is that these reactions are not learned. They are innate, automatic, and deeply wired into the structure of the human brain. Moreover, the idea that pheromones shape our social and emotional lives has been supported by studies on individuals with anosmia, the loss of the sense of smell. People who cannot detect odors often report increased social insecurity, difficulty with intimacy, and even symptoms of depression. This reinforces the idea that smell, and specifically social chemosignaling, plays a key role in emotional regulation and interpersonal connection. In fact, the brain's interpretation of social smells may be as important as visual or auditory social cues in establishing rapport, empathy, and trust. Interestingly, the pharmaceutical and wellness industries have begun to explore these insights for therapeutic use. Synthetic pheromone-like compounds are being developed for applications ranging from anxiety reduction to sexual therapy. However, the science is still in its early stages, and much more research is needed to understand the full range of effects and how these compounds interact with individual biology, context, and expectations. From a behavioral optimization standpoint, awareness of pheromonal communication allows for more conscious environmental and interpersonal design. For example, understanding that stress-related chemosignals can influence group mood has implications for workplace wellness. Managing odor environments through ventilation, personal hygiene, or even scent-infused spaces could impact collaboration, productivity, and stress regulation. Similarly, in high-stakes environments like hospitals, schools, or courtrooms, being mindful of how individuals may chemically influence each other's emotional states could lead to improved outcomes and more humane environments. There is also potential in therapy and trauma recovery. Because chemosignals directly access the limbic system, they could be used to anchor new emotional associations in the brain. For instance, pairing a calming or positive scent with exposure to anxiety, inducing material might help rewire trauma responses through a process called counter-conditioning. This is analogous to how certain smells can trigger powerful memories, only now the process would be guided and therapeutic. Functional MRI and EEG studies are beginning to explore these possibilities, showing changes in neural connectivity and emotional response with scent-based interventions. A particularly intriguing frontier is the potential for pheromone-influenced learning and memory consolidation. Animal studies have shown that exposure to specific social chemosignals during learning tasks can improve performance, suggesting that emotional states, modulated through scent, can influence neuroplasticity. This aligns with broader findings in neuroscience that emotional salience enhances memory encoding. If certain chemosignals can reliably induce brain states associated with focus, calm, or motivation, they could one day be incorporated into tools for education, training, or performance optimization. There is also a genetic and epigenetic layer to consider. Individuals vary in their sensitivity to different pheromones, and some of this variability is linked to genetic polymorphisms in olfactory receptors and hormone systems. Furthermore, early life exposure to specific chemosignals may influence the development of social brain networks, altering sensitivity and responsiveness later in life. This means that chemical communication is not only shaping behavior in the moment, but potentially sculpting the brain's social circuits across the lifespan. Despite the power of pheromones, it's important to emphasize that they do not override all other forms of input. They work in concert with visual, auditory, and contextual cues to guide behavior. What makes them unique is their subtlety and their capacity to operate beneath conscious awareness. Unlike spoken words or facial expressions, pheromones cannot be faked, edited, or hidden by intention. They reflect real-time internal states, making them one of the most honest forms of communication we possess. In sum, pheromones represent a hidden language of the body, one that speaks to the deepest parts of the brain and profoundly shapes behavior, emotion, and social connection. Though still underexplored compared to other sensory systems, the field of human chemosignaling offers rich insights into everything from attraction and attachment to stress regulation and group dynamics. As we continue to uncover the molecular codes that govern these processes, we not only expand our understanding of the brain, but also gain new tools for improving relationships, enhancing well-being, and optimizing human potential. The senses of smell, taste, and pheromone detection do not operate in isolation. They are deeply intertwined within the architecture of the brain, converging in neural hubs that are responsible for assigning value to stimuli, generating emotional responses, and shaping behavior over time. This multisensory integration is not simply a matter of perception. It is the foundational mechanism by which the brain interprets the internal and external world, allowing us to adapt, survive, and thrive within complex environments. When you experience a flavor, what you're actually sensing is the combined input of both taste and smell. This phenomenon is called retronasal olfaction, the passage of volatile molecules from the back of the oral cavity up into the nasal cavity during eating. The olfactory receptors process these molecules in parallel with the taste receptors on the tongue. The brain does not separate these signals. Instead, it fuses them into a single experience, flavor. Without the sense of smell, the perception of flavor collapses. This is why food tastes bland when you have a cold. The taste buds are still functional, but the olfactory input is muted, stripping away most of the nuance. This fusion is managed in the orbitofrontal cortex, a region at the front of the brain just above the eyes, which plays a central role in assigning value, predicting reward, and making decisions. The orbitofrontal cortex receives inputs from both the gustatory and olfactory systems and creates a unified percept. It also receives input from the limbic system, particularly the amygdala and hippocampus, which adds emotional valence and memory associations to the experience. So when you eat a food that tastes amazing, it's not just about molecular binding to receptors. It's about how those signals are interpreted, remembered, and evaluated in the broader context of your emotional and physiological state. What's especially powerful is that this sensory integration influences learning. If a particular food or scent is paired with a strong emotional or physiological outcome, for instance, pleasure, sickness, social bonding, or trauma, the brain tags that combination as meaningful. These multisensory experiences are then stored more robustly in memory and influence future behavior. This is a survival mechanism. It ensures that the organism remembers what to seek and what to avoid, not just based on one input, but based on a constellation of cues. Pheromones add a layer of unconscious, socially derived information into this system. They interact with both the olfactory system and broader emotional processing circuits to guide behaviors that are highly relevant to social interaction. Mating, parenting, group cohesion, and status evaluation. The brain does not process pheromones in isolation either. Their effects are modulated by context, prior learning, and concurrent sensory input. For example, the presence of a pheromonal cue indicating dominance may be interpreted differently depending on visual and auditory cues in the environment. Posture, tone of voice, or facial expression, the integration of all these signals is what ultimately determines the behavioral outcome. Consider how this operates in a real-world setting. Imagine you walk into a room and meet a new person. Your brain instantly begins processing an immense amount of information. Their appearance, voice, facial expression, body language, and scent. You may consciously register their name and facial features, but below the level of awareness, your brain is decoding chemosignals, stress hormones, reproductive hormones, immune profile indicators that alter your perception of them. At the same time, if they are eating or drinking something, or if there's food present, the aromas and taste cues can influence your emotional state and how you interpret social cues. For instance, the scent of familiar food can create feelings of comfort and trust, making you more open and socially responsive. This is the sensory brain at work, using a multi-channel input system to determine the appropriate behavioral and emotional output. Now take that example and apply it to something as complex as mate selection. Research shows that humans unconsciously prefer potential partners who scent signals a genetically different immune system. The combination of olfactory and pheromonal cues informs decisions that are rarely consciously articulated, but have significant biological consequences. Even taste plays a role here. Kissing, for example, is a behavior that may have evolved in part to sample pheromonal and taste cues from a potential mate, helping to evaluate compatibility on a biochemical level. The brain integrates this data to generate a gut feeling, literally and figuratively, about whether someone feels right. From a developmental perspective, this integration begins early in life. Infants recognize and are calmed by the scent and taste of their mother. Breast milk contains both nutritional and chemical cues that signal safety and promote bonding. These cues are processed together: taste from the milk, smell from the mother's skin, and chemosignals from her body, and they shape the infant's early models of trust, attachment, and reward. These early experiences lay the neural groundwork for how the individual will respond to similar sensory cues later in life. It's not just about learning what tastes good, it's about learning what environments and people feel safe, comforting, or threatening. In adolescence and adulthood, this integrated sensory processing becomes more nuanced. The same cues that once signaled maternal safety may now signal attraction, group belonging, or social status. The brain adapts by recalibrating its response to olfactory and gustatory signals based on changing hormonal states and social roles. For example, during puberty, the olfactory system becomes more sensitive to social and sexual chemosignals. This is not accidental. It reflects a biological shift in priorities, from parental bonding to peer interaction and mate seeking. These shifts are regulated by changes in the hypothalamic, pituitary, gonadal axis, which alters not only hormonal output but sensory processing and behavioral responsiveness. The integration of smell, taste, and pheromones also has implications for mental health. People with mood disorders often have altered sensory perception, reduced smell sensitivity, changes in taste preference, or impaired emotional processing of sensory cues. This is not just a symptom, it may also be a contributor. For example, reduced olfactory input to the limbic system can lead to diminished emotional reactivity, a core symptom of depression. On the flip side, overly intense chemosensory perception may contribute to anxiety or sensory processing disorders. Because these systems are so deeply tied to emotional regulation, changes in one domain often affect the others. Treating the sensory system through scent therapy, dietary changes, or environment design can therefore be a way to support emotional resilience and recovery. Multisensory integration also affects cognition, especially decision making and memory. When we learn something in a richly sensory environment with specific smells, tastes, and emotional tones, that learning is more likely to stick. This is because the hippocampus and orbitofrontal cortex tag multisensory experiences as more salient. They recruit more neural pathways during encoding and create more access points for retrieval. This is why nostalgia is often triggered by sensory cues. A scent or flavor brings back not just a memory, but a full emotional state. By intentionally designing environments that include meaningful smells and tastes, we can enhance learning, creativity, and motivation. There is also emerging evidence that these sensory integrations influence group behavior. In social species, including humans, chemosignals can synchronize emotional states across individuals. A shared meal, for instance, doesn't just provide nutrition, it creates a chemically and emotionally synchronized state that enhances group cohesion. The scents and flavors of the meal activate shared reward pathways, while pheromonal cues exchanged during close interaction regulate trust and openness. In this way, communal rituals involving food and scent, such as holidays, religious ceremonies, or family dinners, serve not just cultural but neurological purposes. They calibrate the emotional tone of the group through coordinated sensory input. Understanding these mechanisms allows for intentional modulation of behavior and performance. For instance, using specific scent, taste combinations in classrooms or workspaces can enhance focus or calm. Incorporating personalized sensory cues into therapeutic protocols can accelerate trauma recovery or mood regulation. And recognizing how unconscious chemical signals influence relationships can help people navigate conflict, intimacy, and cooperation with greater awareness and empathy. One of the most exciting areas of research is the development of sensory interventions that leverage these integrated systems for behavior change. For example, pairing a calming scent with a particular food can create a new association that reduces binge eating or emotional cravings. In individuals recovering from addiction, introducing specific taste and olfactory patterns during therapy sessions may help rewire the emotional circuits associated with reward. These strategies rely on the brain's remarkable plasticity, its ability to form and reshape connections between sensory input and behavioral output. This sensory integration is also dynamic. It adapts with experience, learning, age, and context. The brain continually updates how it interprets and weights sensory input based on outcomes. If a food that once brought joy becomes associated with illness or trauma, its sensory profile will change in the brain. The same applies to smells or pheromonal cues associated with people or places. This means we have the capacity to overwrite old sensory associations and form new ones, a powerful tool for personal growth and healing. In essence, the integration of smell, taste, and pheromone signals forms the scaffolding for many of the behaviors we think of as spontaneous, emotional, or social. These systems work in parallel and in unison to inform the brain not only of what is happening around us, but what it means, how it feels, and what we should do next. They influence everything from memory to motivation, from bonding to avoidance, from curiosity to fear. Recognizing their combined power gives us access to a deeper level of self-regulation, social understanding, and cognitive enhancement. By paying attention to these subtle yet profound channels of influence, and by learning to shape them deliberately, we can optimize how we think, feel, relate, and perform. Whether the goal is better mental health, sharper learning, stronger relationships, or simply greater presence in daily life, the sensory pathways of smell, taste, and pheromone signaling offer a direct and transformative route into the core circuits of the human brain.